is to tell you all about the mechanics behind spike sorting, and specifically about spike sorting with Kilosort. Now, I guess you're seeing my screen. All right. I'm having a little difficulty. Here's some of the people that have been involved with this from the beginning. All right, press the wrong button. All right, so my goals today are to tell you about how Kilosort works for spike sorting. After this, Nick is going to tell you how you use it in practice. You're going to have a lot more time to analyze Kilosort results after this. I should point out that this talk has already been recorded more than once, not just at the NeuroPixels course last year, but also on openneuroscience.com with a little more material. So you can rewatch it there. The references for this talk are kind of spread out. The information about how Kilosort works is spread out all over a few papers. Some of the basic mechanics are there in the original NeurIPS paper it's also in bioRxiv. And then if you look at the supplementary materials for these other two papers, you'll find some more information. And then if you look on the GitHub wiki, you'll find some more information. There's no updated unified paper yet, but hopefully there will be soon. And since you can find all of this information, including the talk, online, I'm going to go pretty quickly over some things and just skip some of the more technical parts so that we have time at the end for questions or during, if I remember to check the Q&A. All right, so the need for automated spike sorting isn't new to anyone at this point. There's these probes with very many channels on them that you're here to learn about and learn how to use. There's so many channels that you record so many neurons that it becomes very difficult to manually spike sort these neurons, which is how people used to do it in the past. Let's see. Now, when I was in the Candini-Harris lab, where they were testing these several years ago, and Nick was inserting 8 probes at the same time into the brain, that's when we realized, oh, this is like a really valuable data set that we should really look into. But one of the obstacles preventing us from digging in was that it required a lot of manual curation after spike sorting. And so that really prompted a lot of the developments behind Kilosort, has been to allow us to look at these bigger data sets without all the pain that comes from spike sorting. Okay, I'm going to go through, you've seen data like this today already or yesterday. I'm going to get straight into the mechanics of how it works. This is the Kilosort run GUI, which is the first thing you see when you start Kilosort. Nick is going to tell you a lot more about it today after this, but I bring it up because it's a good way to illustrate especially the pre-processing part that comes before any spike sorting. This tends to be common across many algorithms, not just Kilosort. You load the data in, and assuming you've configured your probe correctly here with the channel map and everything, you will see a picture like this on the right. On the y-axis, here are channels. Every row is a different channel. It's shown as an image. X-axis is time. It's quite zoomed in. Every little event you see here is a spike. So all I believe the red is negative going, black is positive going in this case. And so if you see a lot of these little red marks followed by black recoveries, those are the spikes. And that's the raw data, which in this case with NeuroPixels 1, it's already a little filtered, so you don't see the LFP. Here it is again. And the very first step that we need to do is some spatiotemporal filtering. This means removing the correlations in space and time from the data, because there's a lot of background to this data, as you can see here. This is coming from lots of neurons far from the probe that kind of all talk together. Some of them are closer, but they're too small to be spike sortable. And we want to remove the influence of those as much as possible, leave just the very sparse little events that you see here that almost look like artifacts or noise or something. This is the actual data Kilosort uses, and those little events are in fact standing out over noise quite a bit, and we're able to detect them. After detecting them, we can so-called reconstruct the data using our templates, and you can see, if I go back and forth, you'll see that where some of the artifact-looking spikes were, we've identified the presence of a template from a particular neuron. And if we subtract those two images you saw before, you can see most of the events that were standing out just go away. And of course, making this residual as empty as possible of meaningful patterns is not only Kilosort's job, but is also a sign that it's doing a good job. So if you can still see spikes in here.
There's a question that I think is relevant, which is, what version of Kilosort are you showing right now?
So, good question. And the answer is that this would be common to all versions of Kilosort. So the way that I'm going to go through things now is Kilosort has mostly added stuff to itself over time. Few things from the past have really gone away. And some of the things I'll show you, especially now in the beginning, are so established that not only just Kilosort uses them, but they were even like used before, and just about every algorithm I know of would use this as a first step, this kind of filtering. But for sure, if you start up Kilosort now, you will be, greeted with these kinds of pictures, even in the latest Kilosort 3 version. All right, and yeah, Matteo, please continue stopping me whenever there's a question. It's a little difficult for me to check the Q&A at the same time. Okay, which actually kind of start with a question in mind, here's the kind of pipeline that's common again to all versions of Kilosort. There's some pre-processing, like what we've just seen. Then there's some clustering of events. Well, the pre-processing can include some fairly advanced steps too, like the drift correction, which I'm going to go into quite a bit. There's the clustering. There's the template matching. Once we have waveform templates, it allows us to identify overlapping spikes. And there's some post-processing that happens to detect if units are good, for example. Here's those particular kinds of steps that are involved in all of these. I'm not going to go into all of these steps carefully, just to leave time for questions. Kilosort 1, 2016. This pre-processing step, again, has been retained since then. The details of it you can find in the paper. And now the step that also goes all the way to 2016 from the original Kilosort is a template matching step. And again, there were people that were experimenting with this even before, like Jonathan Pillow and Olivier Marr a few years before. And the idea here is that we will represent our neurons as templates, their spatiotemporal signatures over channels. You can see most of these neurons have like one or a few big deflections where their main spikes are, but also they decay away from that big deflection. That entire spatiotemporal pattern of voltage is useful information. It's something that the spike creates every time it fires. And so we want to integrate that information and build us a good detector based on these templates, assuming we already know what the templates are. We take them, we slide them over the data left to right, convolutionally in time, and we detect places, time points where, one or more of these templates are high, and we subtract off the events at those time points. So let's say, we detected some big events here with our templates. We subtract off a scaled version of those templates so that, we try to make the residual as clean as possible, right? There's nothing left there. And if we do this well, then we subtract one event. It allows us to come back and detect another event that's right next to it. And that's why we say that we're able to detect spikes that are overlapping, to some reasonable extent. If they're right on top of each other, that will be difficult for any algorithm, but it certainly allows us to, identify neurons that we wouldn't have been able to if we had started with a spike detection step. And kind of the demonstration of this is and this is a very old benchmark back from Kilosort 1, but I keep it in my slides just to illustrate this point. So, to ignore the rest, but at that time, here's some template matching algorithms, Python Circus, Kilosort. They're able to miss many fewer spikes. And we think that's exactly because they're able to detect these spikes that are overlapping with other spikes, which these more classical frameworks were at the time not able to. Now, here's a single math equation that characterizes a lot of what goes on in Kilosort. It's a model that we're going to fit to the data. And the model says that if we have a voltage at some channel Z at some time t, that's the data we recorded. We model that based on a superposition, so that's the sum here, over all of our spikes. And each spike has a particular template A that's based on this spikes cluster. So sigma of k tells me for every spike what cluster it's in. This is something we have to determine. And every one of these templates has a particular spatiotemporal profile on its own. And we multiply that with some amplitude, and it gets placed at the times of the spikes. And there's some noise that we assume gets added to this. Now, x here and A and sigma k, the cluster assignments, all of these are unknowns. We have to optimize for them. And so most versions of Kilosort have proceeded as an iterative optimization between finding spike times for given a set of templates and finding the templates given a set of spike times and detecting the amplitude in the mix there too. Now, the main part of this remains the same, which is if I do have the templates, I will be able to find the spike times using the same process. It's just the way we find the templates in Kilosort 3 is a little bit different. And you can ask me about it at the end if you want to know more details. How do I know if it worked on my own data? Well, this is the subject of the rest of the day, so I'm not going to go through it. It involves a lot of phi and looking at neurons in phi, looking at pairs of neurons in phi, even when they're very close together. How do you become an expert spike sorter? Again, this is the subject of the rest of the day, but as far as the Kilosort part goes, this is also something I do to see how well things go. Is I just take one data set that I want to spend a lot of time with, maybe not your best one or your worst one, just a middle data set. You set it up in the GUI, make sure it's set up correctly. You run Kilosort. Now, as soon as you run Kilosort, you want to check the results don't look too funky, which could be a consequence of setting parameters incorrectly in the GUI. So, maybe double check that as a sanity check that things went correctly. And then once you've done that sanity check, now you can go and use the entire capabilities of phi, which is a much more developed GUI than what we have here inside Kilosort. And once you do all of your analysis and visualization in phi, you can come back and fine tune some of the Kilosort parameters that you think. There's just a couple, maybe 3, that are important, and you can try to see how those change your results. And once you've done this quite a bit, that you're happy how it works on this data set, you will ask, you'll just go back and find another data set. I see there's questions accumulating. All right, so I'm going to take questions separately, I think, because just taking them during doesn't seem.
Yeah. Also, you'll be able to type some answers when you're done talking. If you run out of time,
right.
You type some answers.
Good, great, thank you. I shall do that. All right, so that's the approach. Kilosort 2 had one big feature that was important, which was drift tracking. Something you've already heard about drift quite a bit, something that was always important for electrophysiology, and NeuroPixel provides a way to correct for it. There's some other little improvements there. I'm not going to go over the details of, now we had a way to initialize better, just looking at the residual, we could see, oh, there's some stuff left, better initialize a new template there and add it to our ongoing set of templates. That's how we're able to find more neurons and make sure we don't miss as much stuff. There's some of these steps Well, I should describe them because they're useful overall. These are the kinds of mistakes you might see, including in phi. For example, here, like something that were two neurons because they're two different colors, but you can see that really is just one cluster that's been cut into half along this line. And so we have mechanisms inside Kilosort to detect such splits and just merge them back together. So in principle, you shouldn't find things like this in phi anymore, but if you do, we can go back and see, was there a parameter in Kilosort that controlled this kind of splitting process and try to adjust that one. Whereas here you can see the two clusters are very well separated. So we know we're doing a good job there. All right, so assuming these two were merged, how do we decide to split neurons? That's another kind of subroutine in Kilosort. That's really also modeled by the way that we analyze this data in phi when we look at the results. And we know that they're well separated because we can draw this line through this plot, right? That very well separates these two distributions. If we project the two distributions onto the yellow line, we can see, things like this, where we've got these bimodal plots telling us that there's multiple probably sources or clusters along that line. And we make a split decision if this dip in the projection goes low enough. And this is fairly similar to how you'd make a split decision in phi. Now, coming to drift, which I want to spend at least a few minutes on. This is something that you can detect in your data. If, for example, from any kind of feature of your neuron that you're tracking over time, if you can see it's changing quite a lot, like it is here. For these three neurons in the same recording, that tells us this is probably the result of drift, vertical drift, changing the overall amplitude of a unit. Different units are more or less susceptible to the effect of drift. For example, if they're really small, like some of these, like this one here, then you can imagine as the probe moves up and down, this unit is going to start falling in between sites. Other effect of drift, you can see in this case, the amplitude of this neuron goes down until it collapses into the noise floor. And we're only getting a few of this neuron spikes. So this is another case where if you see something like this in your data, you go back and think, oh, can I lower the threshold somehow? Because I don't want to lose this neuron. I don't want to lose the spikes of this neuron at the end of the recording. Okay, and so the solution we had for drift in Kilosort 2 was a tracking solution. I'm not going to go into how we evaluated that was doing something good. It was indeed very helpful at the time, but since then we have, we benchmarked it and we saw it was good. But we actually had a better version of drift correction last year in a kind of intermediate version of Kilosort 2.5. This is the one that went out with the NeuroPixels 2 paper, so you can read a lot about it in the supplementary material of the NeuroPixels 2 paper. And the main feature now is drift correction. Instead of just tracking neurons as they drift, we're able to correct the data a priori. And this is something that's been motivated just by seeing enough recordings, particularly ones that were a little longer, like this one. It's just, it's about 2 hours. You can see there are neurons that are kind of dropping in and out in different places, right? Every color is a neuron. Some of them are tracked for most of the recording, but then they kind of drop out either here at the beginning, like the blue one, or the yellow one. It kind of drops out for half of the session and so on. And so we knew that this kind of failure could happen when you just have a tracking-based algorithm. And at that time, there was enough evidence that would be able to do actual registration of it, just shifting the actual raw data. And so we went for it. Nick actually had a prototype of how to do this for, this is just showing you 2P data that we've had registration for a long time. We knew how to do registration for 2-photon calcium imaging, but we didn't quite know how to do it for electrophysiology. And Nick Steinmetz actually a few years back had a prototype working of how to do this kind of correction for NeoPixels 1 in that case. And we kind of took that and improved it to a point that it was working very well. I'm going to go over, these just some issues of making this comparison with calcium imaging. Yes, it's still registration, but there were some unique issues we had to solve, and then we did. The development of NeoPixels 2 also simplified things a bit for this kind of probe, because now everything was aligned, all the sites, there were just 2 columns instead of 4, basically, no sites missing in between. Gone was the checkerboard geometry. These spikes are really good for data interpolation. So if you can imagine, as the probe is moving up and down, the spikes are moving up and down. We need enough sampling vertically to be able to know what a spike would look like when it's like half shifted between sites, for example. And so if the sites are vertically far from each other, we won't have enough samples. The interpolation won't be able to just produce missing data. Whereas because the sites here are sampled densely enough, it means we have a bit of the smoothness helping us. And the process goes roughly like this. You're gonna probably see plots like this if you haven't already. Do we have a name for these plots? These are some kind of spike
depth raster, maybe
density plot. All right.
Yeah.
It's right. So this is showing like a really long period of time. Every dot here is a spike, and it's been colored by its amplitude. And these were just detected without any kind of spike sorting, just detecting spikes based on some canonical templates that we set up a priori. And what you're seeing through the plots like this is that there's clear visual movements up and down, both at the top of the probe here, at the bottom of the probe. You can see this is an experiment from Nick where he started moving the probe using the manipulator up and down. You can see everything is shifting up and down. And so our goal with drift correction was to make it look like there's straight lines to this plot, right? So to correct the data directly up and down, like literally the raw data, we're going to correct it up and down after detecting how much movement there is at any one particular moment. The details of how we detect that movement, I'm going to skip over. Here's the estimate that we have. So first we have to estimate the movement, and here's the estimate. You can see in this period of manipulator movement, the estimate is pretty good. It looks like that sinusoidal, sawtooth movement that Nick imposed on the probe. And you can see there's also some movements outside of that period, which are just natural movements, not imposed by the experimenter. Now that we know how much the probe moved, we needed an interpolation method. So here's some of the simplest interpolation methods that you can just find. Probably this bicubic one is good enough. It's pretty slow and it's not good with arbitrary geometries. So instead we did a slightly more custom interpolation based on kriging. That's just the name for it. Essentially, if we have any kind of arbitrary geometry like this, then we would like to know what the voltage is at this position here, which we need to, because remember, the probe, we want to shift by, let's say, half a site, or a third of a site or something here. So we'd like to know what the value of the voltage would have been at this position. So for that, we basically calculate correlations of this, not correlations. We set up a Gaussian kernel that says that the voltage should decay smoothly away from the center site. So if it decays smoothly, then it should have, on average, a certain correlation with these sites based on the distance. And that allows us to invert the problem as just standard kriging to predict what the voltage would be here, just based on that smoothness assumption, which is what interpolation is. It's taking advantage of smoothness to determine the value at an unobserved position. Okay, and we can do this for all of the sites as we're imagining the probe needs to be shifted down by a certain amount. And this is the original picture. Here's what it looks after this correction. And so far, I've only shown you rigid registration. That's why that word comes up there. That means we've estimated a single vertical offset for the entire probe, which we know is the bad assumption, but it's a stepping stone towards estimating different amounts of movement in different parts of the probe. And we, just like for calcium imaging, we call that non-rigid registration. We separate, again, very similar strategy to calcium imaging. We're going to divide the probe into shorter segments. I think in this case, there may be like 1 millimeter or maybe 800 microns, something like that. There's 6 maybe segments for a standard probe. And for each one of these segments, we're going to do a rigid registration. Well, just the detection part of detecting for the movement. And then we're going to basically stitch it all back together to have a smoothly varying offset as a function of channel. In the plot here on the right, every color is a different channel, not a different channel group, right? So every color here corresponds to a color here of a group of channels that we've estimated separately. It's Y shift, and you can see that, in fact, for this period where there was no manipulator movement, there's significantly different shifts for the top part of the probe in red and yellow versus the deepest parts of the probe in blue and cyan. You can see the difference can be up to 10, 20 microns or so. And here's that part of manipulator movement again. And now if we just, again, look visually at the results of non-rigid registration. Oh, there's a shift here in the picture. Well, I'll just let you look at this one. And what you should see is it's some of the little movements that we had left here in rigid registration, like at the top, you can see there's a little bit of wiggles left. So, oops, those are mostly gone after doing non-rigid registration. All right. This is what one of those units looks like in Phi for this kind of recording. You can see there's very little noise. This unit is, we've corrected this is after correcting the data. If we didn't correct the data, you can see the same unit has a lot more vertical spread here, and it doesn't look as clean. Here's some of the benchmarks from the paper. You can go look at them yourself in the paper. It basically just says it does a lot of good correcting the data this way. It doesn't even matter what spike sorting strategy you use afterwards, we think. Just correcting the data is a really useful first step. And now here's from that recording, here's some of the neurons tracked for a longer period of time. Here it is in an even longer recording in a freely moving rat. So this is just to show that even chronic implants can have a lot of this vertical motion in rats. Where's my scale bar here? I don't know, but this is on the order of, so these big ones here are on the order of 20, 30 microns or so. So 1 or 2 sites, essentially. And during some periods, there's even like kind of faster motion. Here you can see during the maze running part of the experiment. All right, so I'm getting closer to the end now, which means that I'll be able to take a lot of questions. Here's one part that is actually not, there's a version of it for the Python version of Kilosort, which we've been working on, but haven't yet fully released. And that contains some of these waveform classifiers based, again, on some data from Nick's papers, where he manually curated a lot of units to assign them good, bad, MUA labels. And we can take a fairly standard machine learning classifier, a deep neural network, and train it to do pretty well. So this is just a CNN classifier. There were some other heuristics we compared against here, but just using this classifier on this pre-trained data set gave us a lot of accuracy in terms of predicting good versus MUA units. I'm not going to go over how it works. It's a pretty standard deep neural network. If you combine other sources of information, not just the waveform, but also the autocorrelogram, you can further increase your accuracy. And this is some analysis done by Rajat in the McNaughton lab. All right, so I wanted to end on this picture of what's shown here are like 6 segments of a NeuroPixels probe. This is a visualization you'll be able to do if you run Kilosort 3. And every cluster here that you see with its own color is one detected neuron. You can see in some, there's the XY position is for each one of these segments. So X position and Y have been detected essentially using our new spike detector algorithm in Kilosort 2.5 that doesn't require spike sorting, but it can tell us where the spike is. And so the only information here after spike sorting is the color that I assign to each dot. And this visualization is nice because it gives us a more, it allows us to do something that for calcium imaging, let's say, we've been able to do for a long time, which is just see your neurons in the tissue, where it's a lot harder to see your neuron in electrophysiology because they're only represented by these very sparse events that they fire. And so here's a visualization that allows us to see an entire NeuroPixels probe after spike sorting, and every little blob being one of your neurons. All right, so I also wanted to say that, if you are interested or someone you know is interested to help us further develop methods like this, and particularly at this point, we're interested in people that can establish analysis pipelines. So we have, with Kilosort for spike sorting, but also for other tasks, like for calcium imaging, we have another pipeline. A lot of the difficulty of adopting these pipelines is just establishing initial pipelines. And at this point, we know that kind of, different brain areas, different recording modalities, and so on, requires somewhat tweak the parameters, maybe introduce some additional pre-processing stage or post-processing to just, really take full advantage of these algorithms. And we think someone to develop such pipelines would actually be very useful, not just for labs at Janelia, but also for everyone else who needs to use such pipelines. And with that, I am going to summarize. Here's some of the modules I told you about. Now, at the center of it all, should I say, well, traditionally, we've thought of spike sorting as just this clustering part, because that's where we, assign spikes to clusters and identify our neurons. I think this classical view has, ignored some of the things that we saw were necessary to do good spike sorting. Nonetheless, you still, because clustering is so susceptible to local minima and problems like that, we've had to develop all sorts of split and merge techniques and refinements over the years to get clustering going well. Now, this is the other important problem that we think it's been working pretty well for a while now. The template matching part, that's what allows us to find spikes in the raw data that may be overlapping with other spikes from other neurons in time, so that you're not missing those events, particularly during periods of bursts, of population bursts, when you might have a high concentration of spikes. Now, there are still some brain areas where firing rates can get so high and neurons are packed so closely together that you can have problems that, they can just be too much going on at the same time. It's possible that more advanced, template matching like algorithms could solve those kinds of scenarios, but it's just, you should expect it to be, quite difficult in those kinds of cases. We talked about drift correction a good deal. This is important, especially in head fixed experiments, but as we've seen, even in chronic implants, some chronic implants, I guess I've also seen some that were very stable. It's important to just align the data before we do anything. because having this kind of residual drift in your waveforms can really hinder, especially the clustering step here, because a neuron can just look like multiple neurons as it shifts up and down. Finally, there's all these post-processing steps I wanted to introduce. One of them is just having these unit classifiers that tell us if a unit is good or not. Some other ones I've told you about also have been some of these post hoc merges and splits that we try to do, which would kind of mimic to some extent the operations you would do yourself when you go and try to refine the data in, let's say, Phi, which is the main visualization that has been developed alongside Kilosort by Cyril Rosand for a number of years. You're going to learn all about it the rest of the day today. Just to bring up some of the people that have helped me with this along the years. Okay, so that's basically what the talk part of this. And now I'm just going to start looking through the Q&A, through the questions, and start answering them. All right, so I'm gonna, I guess, read them out loud. How much time do I have now, Nick? Or
I have a lot of time. In fact, we're expecting a break in 19 minutes.
All right, great.
You may decide that you don't want to take 19 minutes and you want to answer some of them, and then the rest you do in typing. You decide how to spend the time.
Well, yeah, I'm gonna go through the questions. I guess it's a little difficult not to have a back and forth with the people asking these, but I will try to simulate that conversation myself. I mean, I did go on purpose over some things that you can just find if you want to watch this talk online. So let's go through by the upvoting. Difference between version 3 and 2.5. Right, so you'd like to know more about this fancy clustering model that we have. So the unit classifier is not yet pushed in Kilosort 3 yet. It's something that we're developing for the Python version because it requires some Python modules, and that itself has been lagging for a while. So let me focus on the clustering model in Kilosort 3. And that's a clustering model that's essentially taking this idea of, well, why did we need a new clustering model, you might ask first? Well, we knew that after we were able to do drift correction and align the data vertically, we knew that now the possibility to use more fancy clustering algorithms were finally kind of opening up, because without that, let me find the plot that I need to show you this. Essentially, this plot. Okay, so you know, in the past, people have used other more complicated clustering algorithms, but we've kind of been sticking with k-means all the way up until, you know, even version 2.5. And the reason for that is that k-means has this, the output is nicely interpretable, because all it does is kind of split the data. You know, data that's elongated in particular dimensions might get split into half, like a unit that's, for example, let's say drifting up and down. They might just produce 3 different templates that track this unit's position along that the vertical axis. And so it made it particularly easy to interpret the results by using a simple algorithm, especially because, you know, we didn't know very well the common sources of problems we might have. Like, we didn't know how much drift was influencing all of this. We knew it had some effect. And so, you know, after we developed these methods to correct the data and remove drift, which I think was the main thing inducing non-stationarities over time, then we knew that, you know, now we were able to probably use more fancy algorithms, particularly the ones that can better deal with local minima and find small neurons, for example, that might not have a lot of spikes. And so in Kilosort 3, the algorithm we developed is essentially an improvement on this splitting approach, where instead of just splitting neurons at the very end as a post-processing step, it's splitting all the way, just starting with all the neurons. This is kind of an iterative hierarchical split of the data. according to these directions that we find that are especially bimodal. Essentially, we maximize this bimodality criterion to make these distributions stand kind of push from each other as much as possible. And finding those splits, we kind of proceed iteratively to until there's no more splits left. And that's essentially how Kilosort 3 works. All right, so that was one question. Now there's another one at the top now. Someone's comparing 2.5 and 3.0. Results are quite different. Some units that had a 14 hertz firing rate, now it has a 17 hertz firing rate. Okay, well, it's not a huge change. The 3 hertz is the first thing I would say. Second thing I would say is it seems like the kind of change where something about the thresholds could be changing, right? So you might want to look at your thresholds. If a neuron's spike rate increases or decreases, it could be because it's hitting this kind of lower ceiling, which I think the default actually is different. The way we treat that lower ceiling is different in Kilosort 2.5 and 3. So it might just be that, which is not something about the algorithm itself. Another possibility, of course, is that the same disputative unit had a 17 hertz firing rate after Kilosort 3 because maybe it was correctly merged back with a little segment of itself that was previously split off. Or because it maybe erroneously added another 3 hertz, firing rate unit. So, the only way to know the kind of problem is to just try to see which one of those options it is. Yes, Matteo?
Well, the question ends with a general question that I feel we should answer. It's important that we get to that point, which is if you're doing a study and in some of your penetrations, some of your recording tracks, you use 2.5, stick to that, right? Or if, don't do half of your study using 2.5 and half of your study using 3. None of these are going to be perfect. Sorry, Marius. But it's more important that you're consistent, right? Otherwise, you might think that some brain region does something and some other brain region does something else, and you're using 2 different pieces of software. Please elaborate. I didn't mean to pontificate, but I think that's an important issue.
Yeah, that's Matteo's point is very good. Yes, indeed. You should definitely only use one version. I didn't see that last question, but it's usually the kind of change you might want to do whenever you want to switch to a new pipeline, right, to analyze your data. You're going to have to put some time into figuring out the differences, like this thing you mentioned, and not focusing just on the one unit here, but really kind of have a broad overview of how things work. And so for that reason, it's not something you want to do mid-project. It's something you want to do, once you've gotten some new data for a new project that you want to get started. You can give this a try. Yes, thank you, Matteo. All right, there's another question. Have we compared with Mountain Sort? Well, Mountain Sort has never run on NeuroPixel's probes, in fact, because it was always a little limited just by memory and computational issues. But there's newer versions, right, from the same people, like Iron Sort is another one. I think it's what it's called, and it's not supported anymore, Mountain Sort either. But you can look into Iron Sort. That's a very new version. I am not really familiar if there's a paper on it or what the details of it are. But that's where I would look. And no, we have not been benchmarking against most other algorithms. The Simon Center people have a website where they run benchmarks against many algorithms. So you can start by looking there. We haven't contributed anything to that. So, take those results with a grain of salt. Those are just the automated runs that they do of the repositories. But that's what I would look. Ability to split the neurons in the feature view has not been enabled in Kilosort 3. Well, that is correct. Yes, it has not been enabled in Kilosort 3. And the reason is that it's some kind of minute work that requires me to focus on, and I haven't had the time to do it. So sorry about that. How well does Kilosort deal with intrinsically bursting neurons? burst detection. Identify bursting cells as single units. So I'm guessing usually behind this question is an assumption that the waveform changes a lot in a bursting neuron from the first spike in a burst, the second, the third, the fourth. And to that, I say, yes, it does change, but I don't think it changes a lot. As far as the algorithms are concerned, we can deal with quite a lot of amplitude variability. This is something that's built into the model. We don't have any other kind of waveform change into the model that would deal with other aspects of the waveform. But I think what's left over there is small enough that most of the time things just work. So for most of these questions, I would say go to your data, identify cases where you think the problem is, in this case, bursting neurons, and then see if indeed the hypothesis you had about it it turns out that it is a problem and should have some kind of special criterion to deal with that. In my experience, that I haven't seen such data yet, even though I always answer this question the way I just did. Difference between template and feature visualization is something that Nick is going to go over when he talks about phi. Cases where Kilosort mistakes changes and overall firing rate for drift. And yeah, there is a good concern to have. I have seen example just like that, but the recording in that case was quite poor. It's not just that the quality of the recording was bad, but it's also the fact that instead of drift correction works really well when you have this long probe like the NeuroPixels probe, and you are sampling like different parts of it throughout the length of the probe, so that you can tell if something moves up and down based on having all those reference points. Now, if you use it in such a way that you're only recording from a fairly short segment, or if your probe, maybe you're not using a NeuroPixels probe, but you're recording from a short segment, you don't have enough vertical spacing to do a good inference of the amount of drift. That's where I've seen it fail. Now, the sleeping part itself, the same neurons are there, they're firing different firing rates. Kilosort won't have a problem with that. But if it's, if enough conditions are bad, like you're recording a short segment, there's a lot of changes in firing rates, then yes, sometimes you'll see things kind of just go completely out of bounds. Like your estimate of drift will just look very noisy and just unrealistic, and you'll know right away that it's not working correctly. Change the selection of electrodes when you record. Well, you'll have to change your channel map, right? That's going to be pretty important. But I think those kinds of details are Nick will know better how to answer them. So please ask him. Track same neurons in different days of recording. Yes, that's another very good question. Yes, Matteo.
Well, you should answer, but I just want to remind people that Anna Lebedeva tomorrow will tell us a little bit. She won't have a lot of time, so if you want to explain how that works, that's great.
Yeah, so just to anticipate a little bit, and then you can ask Anna. But basically, we have a setup where you take two sessions. We know pretty well how to align two sessions to each other. We spike sort them separately, and then we align them. We align the spike sorted results to each other, and that's the one that doesn't work actually.
No, that's not what she does. I mean, she actually pretends it's all one day.
That's right. Yes. So I mean, we've been going back and forth on this because the version that worked the best is in fact the version based on Kilosort 2, which does this kind of continuous tracking of neurons. So yes, Anna pretends it's all one day. It all gets registered to each other with the same drift correction strategy. But now it actually requires Kilosort 2 to kind of process the data sequentially and track neurons over that gap in time between the two recordings. So yeah, things get a little more messy with chronic recording, especially if you have many of them you want to align to each other. So that's still ongoing work that we're trying to fix it for that, but it's quite a bit more difficult.